Hi, Rick.
Oh, this is good. Now
Yeah, so that you can see anybody raise their hand.
Yeah, I can see the audience.
I mean, I don't know how well your microphone will pick up. You might have to yell.
Yeah.
But we're also doing a live stream.
Repeat the question.
Yeah, maybe you can repeat the question.
Sure, I can repeat the question, Rick, and I can sit here in front. And we're just going to.
So, Kathy, this is, I just wanted to show the slide.
Okay, we're also going to try something. Where's your loudspeaker on this?
Kathy, is the slide progressing?
Yes.
Where's it on your computer then? Because I want to see if we can hear it through the microphone.
Can you talk, Rick?
Yes. Can you hear me now? Can you hear me? Okay, I'm on slide 2. Can you see slide 2 going on to slide 3?
Yeah, we can.
Okay.
So do
Okay,
I
perfect.
need to shut off my mic, right?
Yeah,
Is
shut
that
up.
correct?
I am just hearing one audio. I'm not hearing multiple.
Yeah.
Can you hear me now?
You might have to use your mic to hear.
Yeah, I can still hear you.
Okay, great.
Yeah, I can hear you. Yeah. So, Kathy, you want me to stop? You want to stop here at this slide?
Yes.
Okay. All right. So you'll just have to tell me when to progress to the next slide.
Sure.
Yep.
Sure.
We're gonna wait a few more
We
minutes.
should
for
wait.
people
Sure,
to
sure,
show
sure.
how many, but what I'm gonna
Sure.
do is just ask people to sign in, and then there's a little opt out at the end if you don't want anyone to bother you, which is what I usually do. I always do the opt out or unsubscribe, you know. So.
I hope so.
You folks don't have to sign in. It's just because I know who you are already. It's just for others.
I'll send a Slack message.
Yeah, thank you so much. I'll send an email to my lab. I don't have many people in my lab.
Email the GPGG students.
Yeah, email them.
They know about the.
Yes.
Okay. If you guys have trouble hearing, seeing. Will you be talking about cut and run? Let me ask Rick directly, he said talk about cut and run. Rick, can you hear me?
Yes, can you hear me?
Yeah, we can. I have just one quick question before you start. Will you be talking about cut and run with
Yes.
transcription factors? Like I know cut
Yes.
and run works well for histone modifications, but do you have
Yeah,
will you have
Yeah,
any data to present on cut and run with transcription factors?
actually, yes. Yeah.
Okay, great. Oh yeah, yeah, they might have. I just came from the FC class, and then I saw Matt Lane come in, so I might be teaching another class out. Yeah, he's oh, okay. Okay. Are you setting up the schedule for your semester, or should I bring that to her? Purple? You know what's interesting? I put 2 papers on my archive. PubMed now lists them. Yeah, I hate. Yeah. Sorry.
So Rick, we're going to get started.
Okay.
Yeah, so thank you everyone for inviting us here to give you a presentation today. My name's Kathy Stickney, and I've got with me here remotely Rick. Rick, are you okay with getting the slides up?
Yes.
Yeah.
Can you see them?
Yes. Yes, so I'm your local key account manager. I've been working in the Boston area for many, many years. I've known Ala and everyone for quite a few years. I'm excited to be here today. With me I have Rick Sen, who is our field application scientist. He's also our specialist for our Pixel instrument, which is a sonication instrument. And Rick couldn't be here live today because he's doing a wet lab workshop in San Francisco, a 2-day workshop training people on ATAC-seq and cut and run and cut and tag. That's something else that we offer to universities and corporate organizations, these trainings. And so I wanted to, we can go to the next slide, Rick. So this just gives you a quick overview of everything that Active Motif has to offer. And Active Motif is a company that was started 23 years ago by a gentleman by the name of Joe Fernandez. Joe started Invitrogen, which I'm sure you've all heard of. And 23 years ago, Joe sold Invitrogen, and he decided to just work inside the nucleus because that's what he's passionate about. And he's very much a scientist. You can move on to the next slide, Rick. So here on our website are all of the resources we have to you. One that I particularly like is the podcast, because we reach out to researchers all over the world and they tell their story about how they got started in science, what they're doing for research, and a little bit about their personal lives too. You can move on to the next slide, Rick. And so also acknowledgements. A lot of the slides we're showing you today are unpublished work and research that we're currently working on, and so I'm very excited about that. There will be a little bit of information about products too, obviously, but I'm excited with the talk. I've heard it before. It's very rich with scientific information. And the next slide. And so now I'm going to let Rick take over from here. And if you want, Rick, we can keep it kind of an open format, but I'm going to step to the back of the room and let you take over.
Sure. Thank you, Kathy. Thank you to all the hosts for this opportunity. Sorry I couldn't be there. I was in Boston a few weeks back, but then I had to be somewhere else today. And but this is supposed to be very interactive, so please feel free to interrupt, or you can ask your questions at the end. And if there are questions I cannot answer here and now, we will be sure to get those answers to you from our lab. So for people who are new to epigenetics, who want to explore epigenetics, we always start with this slide, which is very relatable because this contains the example of identical twins. We all probably know or have in our family some identical twins or friends where you see when they are much younger, they look more alike And despite being identical, having identical genetics, as they grow older, there are differences that you start to see in them. They start not looking as alike as they were when they were younger. And epigenetics accounts for a lot of that. Epigenetics makes one more prone to diseases versus the other, or more prone to obesity, allergies, infections. So this is just one example which a lot of people can relate to who have not been familiar with epigenetics before. And then for those of us who are familiar with the concept, we know that it is very crucial, very important to our health, development, disease, and there are quite a few FDA-approved drugs that are for epigenetic markers or epigenetic targets. And there is a lot of clinical trials going on across the world which are focused on epigenetic modifiers. So it's very important to understand and learn about epigenetics. And at Active Motif, we have a lot of different solutions to help you design your projects and get the best out of the samples that you have. So we are always there guiding you from end to end, that is from processing the sample throughout the wet lab, and then the sequencing and the final bioinformatics analysis. So that's our major offerings. So one of our major offerings is the services lab, and then we have the whole list of products which Kathy just went over a few slides back. So we are happy to talk to you in details about any of our products and services, and we just don't have to do it all today. If you in the future, if you have something come up, you can always reach out to us. So epigenetics is not just important for biomedical research. Epigenetics is everywhere in nature, as you can see here, including the food that we eat. Even plants have epigenetic regulations, and so things directly impact us every day. So epigenetics is really crucial to understand. And if you look at the complexity of the human epigenome, if you just break down one human into the number of organs, cells, DNA, chromosomes, RNA, you see the numbers just keep multiplying, and there's so much of regulations going on, and epigenetics is controlling all of that. So it's really crucial, because one mistake here can lead to disastrous consequences. And if you take a schematic overview look at this here, so this is the human cell or any eukaryotic cell where you have the chromosomes inside the nucleus. But then if you take a closer look, you'll see the chromosome is actually composed of chromatin, which is a mixture of DNA and proteins. That is a very simple explanation. Now, these DNA and proteins are very complexly, compactly organized into this chromatin structure here. So the chromatin is actually made of these fundamental units called nucleosomes. So the nucleosomes are the protein components. So they are actually an octamer, or 8 histone units, 2 copies of histones, H2A, H2B, H3, and H4. So all those proteins are together, and the DNA wraps around them, 146 base pairs of DNA going around them. And the histones being proteins, they're made of amino acid residues. So the N-terminal histone tails have amino acid residues which undergo post-translational modification. There can be methylation, acetylation, or other components coming in and associating with them or getting away from them. So those post-translational modifications regulate whether the nucleosomes will all come together, leaving no free DNA, no accessible DNA for transcription factors to come in, or whether the nucleosomes will be spaced apart, whether there will be free DNA where transcription factors can come in and cause gene expression. So very important regulation of gene expression is through these modifications, the post-translational modifications of histones. Another way in which epigenetic modifications occur is the DNA methylation. So directly on the DNA, on CPG islands, you can have DNA methylation, which again regulate gene expression. And then there are chromatin remodeling enzymes, which will cause chromatin structure to open up or close. 2 other ways are non-coding RNA. So non-coding RNA will interfere during the RNA translation process, and at the level of proteins or prions. But in today's talk, we will not cover non-coding RNA and prions, but there's a lot of exciting research going on in those directions as well. So this is a diagram which most of you are familiar with if you have taken epigenetics classes, where this is just showing you the breakdown of the chromatin structure here. So these are the histone octamers I was talking about. And these histone modifications you're seeing here are the most well-studied, well-known, but there are many others which are less known, less understood, and many which are not yet discovered. So there's a lot going on which we still have to find out, because those are directly impacting us and future offsprings. Because epigenetic modifications, as you know, they are epi, so it's the Greek word for on top of. So it's the things that are happening on top of your genetics. They are not changing the genetic the sequence, so the DNA sequence is not changing, but everything on top of it is getting affected, and it's heritable. So epigenetics is heritable. Anything that we undergo epigenetically is bound to impact the future generations. Now, the biggest thing, the most fundamental thing that we want to know, that people want to know when they are designing an epigenetics project, is whether there is a change in the chromatin structure, whether epigenetically, whether the chromatin is becoming more open or more closed, because that's like a portal that gives you the gateway into the whole world of epigenetics. So if you are new to the field, then you would first want to know whether the drug that you're testing, or the mutation that you're studying, or the disease state, or the different developmental stages that you're looking at, all of those situations, you want to see whether there is a change epigenetically genome-wide. So whether the genome, or whether the chromatin is getting more relaxed or more condensed, because that will next direct whether the gene expression will go up or down. So the first thing that we want to look at is chromatin accessibility, that's you're seeing on the left. Once you determine whether there is a change in chromatin accessibility, then you want to look at the mechanism. And for the mechanism, you might want to see whether a histone modification of interest is causing that, whether the histone modification is coming in or going out, or if it's an transcription factor, or if it's RNA polymerase II being associated differently in different situations, or if it's a group of proteins, as you can see here on the left, what is changing there. So those are the mechanisms. And then you might also want to look at chromosomal interactions in 3D, because this is another way in which epigenetic regulations occur. So chromosomal interactions are dynamic. They keep changing during stages of development, during other situations, and those will also change the transcription factors that are coming in and playing a role here. So for example, if you are looking at neural progenitor cells being differentiated into the final neurons, then the chromosomal interactions keep changing and the transcription factors keep changing at different stages, and they regulate different sets of genes. So that's very important to understand as well. And then finally, you want to look at the gene expression or the RNA-seq here. Now, people often ask, why not only RNA-seq? Why do we need to study all of these things? Because we are interested in gene expression going up or down. That's because if you don't know the mechanism, you can't target the RNA of your choice to go up or down. So it's important to understand who's the regulator, who is causing those RNAs to go up or down, so that you can target them. Now at Active Motif, as you can see, we have all these solutions. So for ATAC-seq, we have, for chromatin accessibility, we have this assay called ATAC-seq, and I'm going to go over the assays that I mentioned here in the next few slides. So ATAC-seq is an assay that helps you determine genome-wide chromatin accessibility. We have kit and service. Kit is for you to use in the lab, and it gives you, it takes your cells and tissue all the way through to the library. So you get the library at the end, which are ready to go for sequencing. It takes like between 2 to 3 hours for the kit to process your samples, and that's the workshop that Kathy was mentioning about. Then that is bulk ATAC-seq, but then you can also do single-cell ATAC-seq, which we offer as a service in partnership with 10x Genomics on the Chromium platform, and then single-cell multiome, which is ATAC plus RNA together at the single cell level from the same sample. So the same sample you process up to a common step and then split into ATAC versus RNA. And you get both data sets and you compare and integrate between them. Once you're done with the accessibility, then you move on to the mechanism. Now for the mechanism, again, we have kits and services for all of these. So the very important class of assays are ChIP-seq, cut and tag, and cut and run. ChIP-seq has been around for decades. It's very robust, very dependable, and hundreds of antibodies have been validated. So it helps you to determine where genome-wide your transcription factor of interest or histone modification of interest is associating with the chromatin. Cut and tag and cut and run also give you similar readouts, but cut and tag and cut and run have certain differences. One is better for histones, the other one is for transcription factors, and all of those things we will be covering again subsequently. And then there's DNA methylation. You have kits and services to detect DNA methylation genome-wide, and then RIME. RIME is protein interactome. So here, instead of doing a sequencing, we do mass spectrometry analysis. So this is similar to CHIP, where you would first apply your antibody of interest to the cross-linked sonicated samples. Then the antibody will cause immunoprecipitation, and then with that immunoprecipitated sample, you will do mass spectrometry instead of sequencing. So that will give you all the interacting proteins associated with that, your protein of interest in those regions of chromatin. So this is another assay. Now for chromosome interactions, you have HiC, which will give you the 3D interactions of chromosomes, and of course for gene expression, this is the most commonly used method, RNA-seq, and we have kit service as well as single-cell service. So these are the overviews of the different methods that we have and when and how they would fit in to design your project. Now I'm going to go over them one by one, and before that I'll just go over the applications that we or people generally use them for. So mainly you have these. Yeah, is there a question?
There was a sneeze.
Okay, so applications are these, mostly biomedical research, but the last one you see is plant and animal health. So it's a lot of push in the agricultural industry, food industry towards epigenetics. And then people are of course interested in single cell. We are doing single cell, but then there's also a big impetus towards spatial biology, towards multiplexing, multi-histone modifications from the same sample, RNA histone modifications from the same sample, and the samples being very rare, we have to have methods to get things done from the same sample, because they may be liquid biopsy or FFPE, which are very rare samples to obtain from patients. You cannot have the luxury of doing cell culture where you can do like multiple different kinds of assays from multiple samples. So you have to have precise methods which are very sensitive, which can use minimum samples and can do multiplexing assays on all of them. So this is kind of the overview of where we are, and now I'm going to go over how we just categorize all of them here. So this is just what I described in the previous slides. So I'll start with ATAC-seq, which is the first discovery assay which you need to understand before you go on into a big venture of epigenetics methods. So ATAC-seq stands for Assay for Transposase Accessible Chromatin. So transposase is the main enzyme that is used in ATAC-seq. It is TN5 transposase. It's derived from bacteria. At Active Motif, we have the free unloaded TN5 transposase, or in the ATAC-seq kit, we have the transposase loaded with sequencing adapters. So these are universal Illumina sequencing adapters, or the unloaded version that we have, we give the protocol and you can engineer or you can ligate your own adapters. So in the kit, it comes with these adapters, and this enzyme has a property. The fundamental characteristic of this enzyme is tagmentation. So tagmentation is when it causes fragmentation of DNA and tags it with sequencing adapters. So it tags with these adapters. So wherever it finds regions of open chromatin, it's going to go and bind to those regions and cause tagmentation. So schematically, it'll look like this. You'll have this little piece of DNA with the adapters at each end, or you can have a tagmentation here across one nucleosome or across two nucleosomes. So if I try to represent them in a simple manner, it'll look like this and this. So one sample can have all these multiple kinds of tagmented products. You'll have a pool of them. You can have more, you can have longer strings of nucleosomes, but there is a step for size selection in the experiment where we enrich for only these three. Anything beyond is getting excluded. Now, again, further down the experiment, we get rid of all these proteins here, so you just end up with free DNA here, because in the end we will do PCR. So once you do the PCR and the cleanup after the fragmentation, this is how, and you run a tape station, this is how a successful attack-seq tape station looks like. This is the characteristic ski-slope pattern. So you get this peak around 200. This is the DNA with the adapters. Then you get the second one between 3 to 400. And the third one between 5 to 600. Now this is ideal. Often you don't get all these ideal peaks, but at least you should see this first prominent peak around 200, and then the peaks around here. So that's also good. If you have questions, if you are not sure about the peaks you saw, if you're doing it for the first time, please feel free to send those to us, and we can provide comments or help you troubleshoot if it's not something that should be there. So there are ways in which you can address some discrepancies in the peak patterns that you see in the tape station. And then how do you interpret this? So once you get this tape station, then you're happy, you sequence, you do bioinformatics analysis. Then the sequences that you get from the analysis, you will align them to the reference genome, and wherever they form the peaks, those are the regions of open chromatin, because the DNA, the open DNA that has been fragmented down, those are aligning here with the peaks to form the peaks. And where there's no signal, that's closed chromatin. So these are the kind of figures that you are seeing in the publications where they are showing these tracks with peaks. So that this is how you interpret how an attack-seq experiment is done and how the data is analyzed. This was first invented in Dr. Greenleaf's lab at Stanford, and Jason Boone-Rostro was the first author, and this is the first publication. And this great paper has many experiments. One of them I like to show is this one, where they are looking at 3 different drug treatments on this gene. And then what they find here is the treatment with this drug causes attack-seq peaks to show up for IL-2 gene here. This means that the treatment of this drug has caused the chromatin of IL-2 to open up, become accessible to transcription factors. So this is great. Now, how do you determine that, and how do you go to the next step? What should be the next step? So the next step here is, one of the important things people do in an attack-seq experiment is they run a Homer motif analysis. So using the Homer software, they run motif analysis. So motif analysis is when the software finds motifs which in the open regions of chromatin, which are transcription factor binding motifs. So it will computationally give you a list of transcription factors whose motifs have been identified in the regions of the open chromatin that you have detected. And then you can pick the transcription factors of interest and then do an actual experiment like CHIP or something else to see whether there is an actual binding in there. So that's what they did. So the next step, they looked at this candidate, and indeed they found the peaks for CHIP-seq here. So that means this transcription factor is binding right there where the peak is opening up. So the drug caused the chromatin to open up, and then this is one of the factors that is binding in that open region of chromatin. So this is an excellent example to show how you can go from the discovery method attack-seq to the next mechanistic method, that's CHIP-seq, how they complement each other and how you can integrate the 2 data sets to get to your final conclusion. So this is exactly what we are going to do next. I'm going to show you how you can use CHIP-seq, the mechanistic methods, like the CHIP-seq cut attack and pattern run, to get the answers that you're looking for. So they will come right after you have done an attack-seq and you have determined there is a chromatin opening and closing going on. So CHIP, this is chromatin immunoprecipitation. This is the gold standard because all the ENCODE data have been validated by CHIP. Hundreds of antibodies validated. CHIP has been around for decades. A lot of us have done CHIP. So how this works for people who are new is you harvest your cells or your tissues, then you have to perform formaldehyde-based cross-linking. Sometimes you have to do double cross-linking. And then sonication. Why? Because your pieces of chromatin are super long. You cannot work with those long pieces of chromatin or DNA. You have to get them to workable sizes. That is, for CHIP, the ideal is between 300 to 400 base pairs. So you sonicate them, and then with those pieces of DNA, you treat them with the antibody of your choice, which go and bind to your protein of interest, and then that is immunoprecipitated, and you purify all those immunoprecipitated DNA, do library prep and sequence and analyze. And anything that is non-specific is washed away. So you can appreciate that it is very crucial to have a good antibody for CHIP. The antibody must be very specific, no off targets, no non-specific, and no backgrounds, because only then you will get your successful pieces of DNA in the experiment, because otherwise you'll get a completely wrong interpretation. The second most crucial thing is sonication, because if your pieces of DNA are not workable sizes, not optimum sizes, then they won't be immunoprecipitated properly, and that would again lead to a wrong or a failed experiment. So these two are very crucial things to keep in mind for CHIP, and for sonication specifically, we have the solution of Pixel. So I'll just take a moment to go over Pixel and then go back to the chip. So this is an instrument which we are happy to do a demo if you are interested in doing chip, DNA methylation, high-C, protein work, LCMS, RNA-seq, or any mesoscale discovery, any kind of thing. Very user-friendly and economical in the long run, and all these kinds of samples have been processed by Pixel. So just something to keep in mind. Now coming back to chip, so here's,
There's a quick question.
Yes.
What's the difference between Pixel and a bioraptor? Because we have a
The
bioraptor.
throughput? Sure, yeah, the throughput. So Pixel lets you process 1 to 96 samples in the same run. Bioraptor the throughput is I think up to 12, 16, or 18. And then the cost of the labware is proprietary for Bioraptor, but we don't have proprietary labware. So you can just get the 96 well plate from Corning and process your samples. And the setup time is of just a few minutes because everything is auto-cooled and regulated, and we don't have water, so no degassing and all this. So, and the biggest thing is consistency. So there is a publication that shows a peer-reviewed publication in nucleic acids research, which shows the consistency. And a lot of customers have also vouched for the consistency of Pixel. And it's very versatile. As you can see, it can process these kinds of samples, fits into these kinds of workflows, and fast.
Okay, thanks.
Thank you. So, chip workflow. You got your samples cross-linked, sonicated. Now here is all your samples. So you have these pieces of DNA with different kinds of proteins bound to the different pieces. Now suppose you want to look at MYC binding genome-wide. So this is the antibody you give, and it binds to the green MYC protein here. So these loci should be the ones that are finally analyzed. So they are immobilized on beads, and the non-specific are discarded. Then you purify those DNA, and then when you map them to the reference genome, the peaks you see are the ones which are pulled down by the antibody. So this is the schematic of the workflow, how you analyze and interpret chip results. And the reason I'm showing this is because there will be a lot of slides coming up next where you'll see these kinds of chip peaks. So in case you're not familiar with looking at chip results here. So now comes the new part. Chip is expensive. Chip is great, but chip is expensive, and chip requires a lot of sample input content. So you need sample content in the number of millions. So for those of you who have done, you'll know that it's like for transcription factors, Active Motif recommends 5 million cells per sample. For histone modifications or RNA polymerase II, which are more robust, it may be 1 million. So we have come up with something that can address those. And also the antibody, the content of antibody you need for chip experiment. If you are doing high throughput, multiply that by 96. And the sequencing depth that you need for chip is greater than a lot of other assays. So again, you multiply that by 96. And I'm just using 96 as an example of high throughput here. So to address all of these, we developed this chip technology, which is for tagmented, indexed, pooled ChIP-seq. Now what is this? This, as the name suggests, tagmented, it uses the enzyme TN5 transposase. So here, instead of traditional sonication of cross-linked samples, we subject those cross-linked samples to TN5 tagmentation. It's exactly the same as a tag-seq. So it undergoes tagmentation here. We have this plate, a 96 well plate, where each well contains the TN5 enzyme and each of those have unique adapters with it. So you have 96 uniquely unique adapters in the TN5s of each well here. Then you add your samples to each well. So maybe you got 96 different patient samples and you're adding them to each well. Now, schematically, each sample will look like this after the successful tagmentation. So you have unique adapters for each. What this lets you do in the next step is it lets you pool all of them in one single tube because they are all uniquely barcoded. From here, from this one tube, you can take aliquots. This is showing 3, but we can actually support 4. And you add your antibody of interest. And then you proceed like a chip reaction. So overview is here. You have your samples prepared, 96 samples. Then you add them to the plate containing TN5 uniquely barcoded up to 96. They undergo tagmentation. You use the pixel not for traditional sonication here, just to get the lysates out, few minutes. And then combine all in one single tube, take aliquots, add your antibody. When you add your antibody and subject them to immunoprecipitation, then with the content of one sample and one antibody, you are actually doing 96 simultaneous chip experiments in one single reaction tube. Then you process that one experiment as just as one experiment throughout. You take it all through the process of sequencing. Finally, during the analysis, you can identify using the unique barcodes which signal is from which sample. So you can do up to 4 different reactions. So this is the overview of TIP-CHIP. Now, I'm very excited to present this. We are very excited to present this, but we are still at the R&D stage of this. So this is not yet completely launched, but we will be launching this soon. And at that time, we will definitely publicize this to you. Now, the first question people ask for this is using tagmentation, the TN5 transposase. So is this biased for just open chromatin? Can I look at heterochromatin associated proteins or histone marks using TIP-CHIP? So for that, we have shown this experiment which rules out no ATAC bias for open chromatin. The bottom 2 tracks, the blue and the red, are for the fixed cell and native cell attack seq, which shows that those are the regular attack seeds and they have peaks for open chromatin. But in TIP-CHIP, there is a proprietary step in the method where we address that and there is no peak for open chromatin. You see a uniform lawn of signal for throughout. So it is as good for open chromatin as for closed chromatin. And the next slide here shows that. So if you look at the first one here, this is comparison between ENCODE and TIP-CHIP. So this H3K27 acetylation is a transcription activation mark, and K27 trimethyl is a repression mark. But you see that you can get signals for both of them. So this is not just for open chromatin. The second thing to show here is we are showing the comparison between TIP-CHIP and ENCODE. ENCODE is what is previously published up there, which we all globally people use as a standard. You see the signals are very nicely matching with each other. Now you will ask, why is our signal, the intensity is different versus ENCODE? So there are 2 reasons. One is we did not do the ENCODE experiments, so we did not control the cell numbers that they use versus ours are different. Also the bioinformatics pipelines, methods of analysis, those are as well different from ours. But the takeaway from here is that the patterns are very nicely matching with each other, despite the differences in the intensity. Now coming back to the TIP-CHIP parameters, what I will be showing, what I have shown in the next slide, and I'll be showing in the other slides, is just for 12 experiments, because we are in the stage of like 12, 24, and going to 48, and before we do the 96. So the 12 samples are pooled, and you can see 30 million reads for each sample would be done in a traditional ChIP-seq, but here we just did one bulk sample for 30 million reads. So cutting down the cost of sequencing. This is the sequencing parameters. Cell numbers. Instead of million cells, this is just 100,000 cells per sample because we pool them all together. And the active motif chip validation protocol has antibody recommendations, so we just use the antibody recommendation for one experiment there, depending on the kind of antibody. Then these are other histone modifications we've looked at, RNA polymerase II, comparing TIP-CHIP with ENCODE, and further other ones. Now, you may not have high throughput to do every day, but you would still want to use less samples and less cost if possible. So for that, there are cut and tag and cut and run methods. These were developed by Dr. Stephen Hanikoff at Fred Hutch in Seattle, Washington. Now, cut and tag, it addresses some of the disadvantages like, the sample requirement and the depth of sequencing and so on. So cut and tag is for cleavage under targets and tagmentation. Again, tagmentation means it uses the protein A TN5 transposase enzyme. So in this case here, we do not need to do cross-linking. You just process your samples, some buffer treatments and some steps, and then finally you come to the main step where you do the target-specific primary antibody. Then you add a secondary antibody which will bind to the primary, and then you will add the TN5 transposase. So this TN5 transposase is conjugated to protein A, which will bind to the IgG of the secondary antibody, and the TN5 will cause tagmentation activated by magnesium. And then this is your immune complex. So because this is tagmentation, you're finally getting a tagmented DNA with the library, which is the adapters at each end. So you're getting the final library at the end of the cut and tag experiment. This is great, but it is limited to histone modifications mainly because of high salt washes that are unfavorable for transcription factor binding. These are the list of antibodies. You'll find them on our website, which we have validated for cut and tag, and we are continuing to expand this list. You'll see mostly histone modifications and some robust factors like RNA polymerase II, CTCF. So this was Dr. Hanikoff's first paper on cut and tag, and this shows very nicely that there is much higher resolution and better signal-to-noise ratio in cut and tag compared to other methods. And if you're not familiar with this protocols.io, this is a website where he maintains all his protocols, and he has published much more with cut and tag, including various different kinds of protocol modifications, sample types, and also you'll find a lot of resources there. And he actively answers people's questions here, so he encourages people to reach out, and he is happy to help them. So if you have questions, you can ask us or you can ask him. So the next one we have is cut and run. So cut and run is cleavage under targets and release using nuclease. So it's similar to cut and tag, except you don't have a TN5, you use a microbuccal endonuclease enzyme, which is conjugated to protein AG, and that is activated by calcium. And what you release here at the end of the experiment is just the DNA, because it's endonuclease, it's not transposase. So you have to do an additional library preparation step, but this has a greater scope for transcription factors for antibodies. These are our list so far, and we are expanding on this right now. Now, there have been situations where people have done mild cross-linking and been able to be successful with cut and tag for a greater range of transcription factors. So if you are currently facing challenges with cut and run for transcription factors, I would encourage you to reach out to us, and we can help you with some protocol optimizations there. Now, because these assays are new, here's a comparison with CHIP and with ATAC-seq, and you can see that they are all very nicely corresponding with each other. Now, this table is from our website, and this is a summary of between the three methods, and it shows you when and why you can use one over the other. So the first thing is the antibody. If your antibody is not being able to be validated after various, you try various kinds of troubleshooting and all, you still can, then of course ChIP-seq would be the way to go. But if you can validate that, then you can choose between cut and tag or cut and run. Now, depending on if it's a transcription factor, maybe you want to go with cut and run. You can see the cell number content requirement, the read depth. Those are challenges that people have in terms of costs and availability. we are currently doing a services grant competition. So we do them from time to time. Earlier it was single cell, so this time it's cut and run. And Kathy can provide you more information on this, but if you have a project on cut and run, we encourage you to submit abstracts and we will do the project for you end to end. So all wet lab sequencing and bioinformatics, there are 3 prizes. The abstract deadline is October 31, and this is on our webpage, our homepage. You'll see if you log in there, you'll find see there, and you'll have to submit an abstract by the deadline. So far, what I presented to you was the regular assays, but then we had to come up with something to address situations where we could not see global changes which we were expecting. And the bioinformatics alone was not able to detect those changes. And for that, we came up with this method called spike-in. It was a collaboration between us, Genentech and Constellation, and Brian Egan, who's the first author on this paper. He recently did a webinar which will give you the detailed description of the spike-in, and it's very interesting to listen to. Because of time constraint on this event today, I will just go over briefly so why we do spike-in and when we need to do spike-in. So it started with this very well-known method, very well-known phenomena of EZH2 enzyme promoting histone H3K27 and trimethylation. And if you inhibit EZH2, then this trimethylation signal goes down. This is exactly what we saw with this Western blot, 2 different inhibitor treatments giving you the same results, which is very much expected. However, when we did ChIP-seq under the same conditions, we don't see the decrease in the signals for these 2 situations here. Hence, we came up with spike-in. So spike-in is when you have your experimental sample in ChIP-seq, so you have your chromatin and your antibody. You spike that with Drosophila chromatin and Drosophila specific histone H2A variant antibody. You put all of them in one single tube and they undergo ChIP reaction. And you process them as one single sample. Now, in our manuals, we make specific recommendations as to the ratio of the human or the experimental versus the Drosophila content that you will be adding. So there are concerns from the field that what if my spike-in content is more, I get more spike-in reads and all that. So we like to say that probably what you're thinking you're adding in the experiment is inaccurate because of various reasons. So in our kits or in our services, we make very accurate recommendations and calculations for you, so you can just follow that, and then you won't be facing those situations. And the second thing is cross-reactivity between antibodies. So we've validated this several times. We have several customer publications on ChIP-seq spike-in where people have published them. So there is no cross-reactivity that we have seen here. So you process them as one single sample, and then finally during the sequencing analysis, you map human reads to the human genome, Drosophila to the Drosophila genome, do a calculation for normalization, and then you get your results. So if you are interested, when you go to our products page, this is where you'll find all the spike-in associated products, and the manuals will give you the calculation of the content to be added and the normalization.
Rick, I have a question.
Yes.
Can you flip this around and use human as a spike-in if you're targeting a Drosophila experiment?
I mean, in theory, I don't see why not, but we will somebody has to do the experiment.
Okay. Yeah. No, but we study Drosophila.
Oh, great. Okay.
So I'm wondering if we could flip the experiment around, use human chromatin as a spike-in to normalize against Drosophila chip.
I think that would be a great experiment to try. Yeah, because all our spike-in are based on the S2 cells of Drosophila, so I don't see why not the other way. Yeah.
Okay.
But if I'll bring this up with the lab, if I get additional recommendations, I'll let you know.
Okay, thank you.
Sure. So this is the calculation you'll find in those manuals if you're interested how normalization is done. But after normalization, here is how the signals look. So now you see this is reflecting what you saw in the Western blot. So this was very nice to see. So recently, earlier this year, we launched spike-in for attack, cut and tag, and cut and run. I won't go over them in details, but it's just to show you that we have these options as well. And for cut attack, it's just the nuclei. So when you start your experiment with your experimental cells, you add the Drosophila nuclei. For cut and tag and cut and run, you add the nuclei with your Drosophila specific antibody. And then this star means it's undergoing the same regular attack process or these processes, and then all the way up to the sequencing, and during analysis, you just map them to the respective genomes. So spiking for attack-seq is to show you that there is a decrease in signal if you reduce the cell number. So on the left, in the x-axis here, you're looking at reducing cell number titration. So your signal intensity is expected to go down, which you don't see as much. But then if you do spiking, you see this is going down. So this was a proxy experiment using cell titration. What if you do a real drug treatment? This is interesting, because here we are doing the same drug treatment against EZH2 to inhibit the repressive transcription mark. So if you repress transcription, what happens? Transcription activation happens, chromatin opens up. So your attack-seq peak comes up. This is unnormalized, no spike in here. This is regular. So here you did the inhibitor treatment and your attack-seq peak is opening up. So this is expected, this is good. What happens if you do spike in? Nothing happens here. That's because at the bottom you see the tracks. This is a localized Hox gene cluster. So this change is a local change. This is not a global change. So spike in did not affect anything differently here. It is the same as with or without spike in. So this just confirms this is a local change. But in the previous cell proxy thing, that was a global change, and that was where spike in is needed. So these are situations in which you will need spike in. This is just an control gap DH where there is no change for anything. Now for cut and tag, here is the histone modification, the very same one which you inhibit with GSK126. Indeed, with spike in, you see the signal is down versus no spike in. Now, if you are concerned that the spike in will give you off-target effects, it will give you low signals where you should not see low signals, we did this experiment to rule that out. We looked at K9-trimethyl. Previously, people have published that this is not affected by GSK126. Using our method, we see there is no effect here. So spike-in has no off-target effects. Now coming to cut and run. So this is where I'm going to show transcription factor cut and run. We chose these two, EED and P53, because they are also part of that same K27-trimethyl, the whole process here. So the inhibition of this, does it affect the transcription factor binding by cut and run? So the first here you're seeing no spike in. So for both, these are how the signals look like with and without treatment of inhibitor. With spike in, you see the signals are going down, much down compared to what you see. So this is transcription factor cut and run with spike in for these things. Now apart from DNA, histone modifications or proteins, cut and tag also helps you detect R loops. This is something we developed. So R loops are RNA DNA hybrid. Oh yes, yeah.
Yeah, we have a request. We're wondering if you could do a quick overview of RIME.
Finish the R loop thing.
Yeah.
RIME?
Finish the R loop and then skip to RIME.
Okay, sure. Yeah, sure. So if you're interested in looking at R loops, because they are associated with transcription start sites, and a lot of pharma companies are focused on them because associated with abnormal gene expression in cancer, there were complicated methods to study this. So we simplified that by adapting our cut and tag for R loops. So that's one. And we validated that in 6 different cell lines that is working perfectly fine. And the next is we did spike in for R loops. Again, the cell proxy showing that spike in gives you the true signals. And then inhibitor treatment is again showing you this. So yeah, that's, and then the one other thing before I go on to RIME is this. The second importance of spike in is to see the true signals for negative control. So the first one we saw was just global changes in decreasing, increasing signals. But here is the second thing where you did not see the decrease in the negative control for RNase A. RNase A should have completely digested your R loop. So with spike in, you see this. Okay, so now I'm going to go to the RIME. So Okay, and this is just our cell-free DNA methylation for 5 nanograms input. Okay, so let me stop sharing this and bring up the website here. Okay, let me know if you can see my screen.
Not yet.
Okay, can you see it now?
Yes.
Okay, great. So RIME is a service that we provide for protein interactome. Here, this is the overview. So cross-link your samples. So this first part is just like CHIP. You isolate the nuclei and then you sonicate. Once you sonicate, then you add your antibody of interest and you immunoprecipitate that. And then you perform mass spectrometry to identify all the proteins that are interacting with that region. So instead of doing a sequencing like CHIP, you will do that, and then you can integrate that with a ChIP-seq experiment to compare to see where, how the ChIP-seq turns out and how for that specific antibody and how the protein interactome is. So people usually do both, or you can do one as a project. So that's the overview of RIME.
But how do we do it if we have a custom antibody to a transcription factor? What do we do we do the RIME ourselves and send you the final IP target and then you guys do the mass spec, or how does this service work?
No, it's end to end. So you would so first of all, the antibody that has, and these are all our RIME publications. So here's the sample preparation. You would have to cross-link your sample if they are cells and send to us, and if it's tissue, I think it's frozen. So we will do everything. And even if the antibody is successful for ChIP-seq, it's not guaranteed that it'll be successful for RIME or cut and run or anything. So for every new assay where we have not done validation for that specific antibody, we always start with a validation. So you will either send us the antibody, or if it's from a vendor, you can tell us, we can source it from a vendor, and then we will do one sample validation. And then based on the success, we can take the full-scale project.
Can we send you our demonstration that the ChIP-seq worked really well? And so we just send you cells and our antibody, and then you can go forward, or is it a must that you validate with your own ChIP-seq?
What stage are you wanting to send us? Like what stage of the experiment?
We want to identify the novel proteins associated with this transcription factor. We've already done ChIP-seq, and it worked really well. We don't know what's the cofactor interacting with this transcription factor.
Yeah. Okay, so we can do the RIME, and then you can so you want to integrate your ChIP-seq results with the RIME results. And in that case, we can check with our bioinformatics team if they can. I think they should be able to do the integration, even though the ChIP-seq has been done different, by you. But if not, we will give you the RIME results, and you can get it integrated and analyzed with your bioinformatics team if you have one. So we can do either way, but I think we should be able to integrate your data sets with our RIME as well.
Okay, great.
So yeah, it's just a high content of cells that you will need, and fix like ChIP here. Yeah, and tissue is just freezing. Tissue doesn't need to be cross-linked or anything. So we can share all these with you.
What's the typical cost of a RIME service?
It's between like 2,500 to 3,000 per sample. Kathy, is that right?
2,500 to 3,000?
Between there, I think.
That's not horrible. Okay. Anybody else have questions? This is your chance to ask?
Were there questions about
Ron, I'm curious what the readout for the rhyme looks like. I think you showed us peaks, but can you give us example data of what you get from the rhyme?
Yes, yes. I'll have to go into this. We have like a whole set of, like what do you call the file? So you'll have to download this FileZilla, and we can share login with you where you can see our sample data sets. So you can, it's not only rhyme, but everything else that's in all the methods that we offer, so you can get an idea of all the deliverables that we have. So, and I'm just trying to pull up something here.
So right now, your promotion, Kathy, is it just for cut and tag, or is there a similar type of grant thing for Ryan? Just for cut and tag for now? Okay.
Yeah. Okay. Okay, here. Sorry. So I'm just showing you one customer publication that was on our website here. Here, can you see this? Figure 5. So yeah, this is the peptide coverages.
You're not limited to specific species. You can do it any species with a proteome.
Let me check that. I know for a while it was just human and mouse, but let's see here. I'll look that up, but I know it was for like. Are you looking for Drosophila specifically?
Yeah, Drosophila.
Okay, I'll check that. I think it was like just human and mouse for a long time. So.
Okay. Any other questions?
Are there any other questions?
I think this is great, Rick. Thank you very much.
Thank you.
Thank you. Thank you so much for attending and for hosting. Bye.
Great. Thanks, Kathy.
Yeah, thank you. Yeah.
I'm actually quite interested in the rhyme.
Yeah.
You can help me look into.
And I'm also going to send you the logon information to download the data samples so you can send them to.
Okay.
I don't.
Yeah, I didn't even know about this method.
Well,
The
yeah,
rhyme.
the rhyme
Rhyme's
is.
really cool. I have a customer right now. Yeah, I've got a customer that's doing transcription factor chip seed. She's
Yeah.
just getting her data.